Today, I'd like to thank both Kevin and Dolores for inviting me here. I'm going to present the work of our group, and it is a village. It's a large group of individuals, a couple of 30, 40 people at least, who have been focusing for the past 15 or 20 years on developing a vaccine for human malaria. In fact, in particular, pediatric malaria. I'm often asked, why do I focus on malaria? Why malaria? And it's usually from one of my snarky ex-physician scientist student colleagues, and they go, Jake, why don't you work on something really important, like male pattern baldness? Or they remind me of those other diseases that middle-aged men get, and we have to listen to the ads for their treatments on Sunday football. Why don't I work on those things? And as I get older, I'm starting to think maybe I should change my focus. But no. No. We work on malaria, and the reasons are really both simple and compelling. We work on malaria because it is the greatest single-agent killer of children on the planet, bar none. Nothing comes close. We're talking about, at the sort of rosy-colored estimate, 500,000 individuals a year. At the more pessimistic side, 2.5 million individuals every single year die of malaria. The vast majority of those deaths are in children under the age of 5, and they're concentrated in places like sub-Saharan Africa and Southeast Asia. So it's the great culling machine of the tropical world. And so at least 8% of all childhood deaths on the planet are due to malaria. So that's really the reason we focus on it. Again, it's the most important single-agent killer of children. So the health economist Jeffrey Sachs has coined the phrase, where malaria has prospered, people have not. And this is a map of the malarious world, and it's color-coded. The warmer colors, the reds and oranges and yellows, are where malaria is rampant. And what you can see is that it's concentrated in sub-Saharan Africa, Southeast Asia, and parts of Latin America. There is no question in my mind that the correlation between political stability, economic development, capitalizing on what might be limited educational opportunities, all three of these social inequalities are concentrated where malaria is doing its killing. It's impossible for me to imagine a community where you have an under 5 mortality rate of 25%. That's 1 in 4 children not making it to their 5th birthday. It's impossible for me to imagine a community like that thriving. And so those are really the reasons that we're focused on malaria. If you're going to go into battle with an adversary that has been defeating you for millennia, you best understand its entire strategy. What are its weaknesses? It will behoove you to understand your enemy in order to combat it effectively. And so unfortunately, and if there's anyone in the room who's suffered through medical school, they're like, oh Christ, not another life cycle. We have to understand how this parasite works. And so it begins with the bite of a female Anopheline mosquito. Every mosquito bite you've ever gotten has been a female mosquito. The females need to take blood in order to lay their own eggs. And so it begins with the bite of a female mosquito. Any of you guys see the movie Aliens? Sigourney Weaver? The first and the second one, the third, by the fourth one, it was, This parasite is as well adapted as that maw crusher. It is so smart. It has been forged in the crucible of evolution. It knows that the first thing that mosquito is going to do is what when she bites you? Is it going to suck? No. If it did that, blood would clot in the straw and she would starve to death. The first thing that mosquito does is it spits. It is anticoagulating the straw, the proboscis, the drinking thing. Where do you think the parasite is hiding inside the mosquito? In the salivary glands. Isn't that crazy? It's in the salivary glands, she spits it in. The form that she spits in is called a sporozoid. That sporozoid is, again, an amazing killing machine. It is in your bloodstream for no more than 5 minutes because it knows your immune system is ready to get it. It's 5 minutes. 5 minutes. In 5 minutes, what it's done is it's sniffed out your liver. This is supposed to be an artist's rendition of a liver. I need to get those holograms, don't you think? This isn't good. So that is a liver. She spits in about 30 of these blue sporozoids. They're tiny. I'll show you a picture of them in a second. In 5 minutes, they've found your liver cells, and each one has infected a liver cell. Now, I know what you're thinking, oh my God, this is going to be a disease of liver. It's going to be a liver disease. No, not true. I tell the students at Brown, the undergraduates especially, the medical students not so much, but the undergraduates for sure, that they'll do more damage to their liver on the average Friday night than will occur in all of sub-Saharan Africa due to malaria. So in go the sporozoites, each one infecting a liver cell. No disease. 2 to 4 weeks go by. For the malaria that does most of the killing of humans, it's called falciparum malaria. 2 to 4 weeks go by completely silent. During that time, the aliens are breeding. Each sporozoite is undergoing massive rounds of asexual reproduction, making these baby parasites that are called merozoites. One sporozoite went in, and up to 10,000 merozoites are jam-packed in that cell until that cell can't keep them in anymore, and it, just like on aliens, explodes. And out come 10,000 merozoites. Still no disease. The next thing that happens, these merozoites infect red blood cells. I have to tell you a little bit about red blood cells. So red blood cells are the cells in your blood that deliver oxygen to your tissues. So they're mission critical for delivering oxygen to your favorite organ: brain, lung, gut, placenta if you're pregnant, kidney. If you don't have enough healthy red blood cells, you can't get oxygen to those tissues, and those tissues die. So that's quite bad. So here's what happens. One merozoite goes in, infects that red blood cell, and now another wave of asexual reproduction takes place. This cycle takes about 2 days. Merozoite goes in, and it becomes something, we call it a ring form. Doesn't that look like a college signet ring? It becomes a ring form. There's some other forms, trophozoites and schizonts in here. It's multiplying. And at the end, one goes in and about 25 baby merozoites, jam-packing that red cell again, and then that red cell shatters. And when that red cell shatters, 25 merozoites come out, and each one infects again and again and again. If any of you are old enough to remember that Joe Namath commercial for Fabergé Organic shampoo, remember, you tell two friends, and raise your hand if you're old enough to remember that. They tell two friends, and they tell two friends. That's what's going on here. You've got geometric progression of the parasites going up, but what else do you have? Geometric decline of the red cells going down. You become more and more anemic until you can't deliver oxygen. One more little curveball, the infected red blood cells are sticky. They're sticky like they have Velcro on them. And that Velcro, those Velcro molecules, stick in the periphery in your vasculature. And so you get clogs. It's kind of like when your kid flushes their socks down the toilet, it clogs the drain. And if the drain gets clogged, fresh blood can't get past the clog, and so the tissue downstream that's supposed to be getting oxygen doesn't get it. So that's all going on here. That's where the disease is. Every once in a while, the parasite gets sick of having all this asexual reproduction. They decide to get it on, and they differentiate into male and female gametocytes. Only they can tell themselves apart. But if they both get taken up by another mosquito, they find it so romantic in the belly of the beast. They conjoin, they form a fertilized egg, and this egg mutates and grows up inside the belly of the beast, up through the guts, up through the thorax, up through the chest, until it's developed into sporozoites in the salivary glands again. So that's what completes the life cycle. That's all bad news. Some good news. There are people trying to develop vaccines that block sporozoites from entering the liver. They've got 5 minutes in order to stop that parasite. It's hard. There are people trying to block merozoites from infecting red blood cells. The merozoites outside the red blood cell for only 140 microseconds. So that's a challenge. Our group is focusing on trapping the merozoites inside the red blood cell so they can't get out. It's like being trapped in a burning house. God forbid there's a fire in here, you'd be trapped. There are people that are trying to block the hanky-panky and so prevent the transmission, which I think is totally cool. So I'm going to tell you about a vaccine that tries to block the red blood cells from getting out. But first I want to tell you a little bit about the misery of malaria. This is what a sporozoite looks like. These are the thread-like forms that she injects into your body. This is a blood film. This is the tool we use to decide if someone has malaria and how much malaria do they have. If we collect enough blood films on you for enough periods of time, we can decide, are you resistant to malaria or not resistant to malaria, based on how many parasites you have. These are healthy red blood cells. That's what delivers oxygen. These are infected red blood cells, and what you can tell immediately is some of the parasites look sinister, and you can also tell It took forever to find this, by the way. And I'm a pathologist. I use the... I'm working it. So, but on a serious note, half of these red blood cells are going to burst in the next 48 hours. How do you think that's going to feel? It's a disaster waiting to happen. So this is actually why I got into malaria in the first place. This is a disease that I got when I was 18 years old, and it really galvanized my interest in malaria. I was lucky enough to survive it. If your picture is up here on a slide, it means you didn't survive it, unfortunately. This is brain, and this is brain from a child who died of cerebral malaria. This is supposed to be a capillary, which is a small blood vessel that red blood cells need to pass through in order to deliver oxygen to the brain. And what you can see, remember I told you that the infected red cells are sticky? All of these things are infected red blood cells, and they've clogged up that artery. No Drano would be able to clear it, which means no good blood can get past to deliver oxygen to this tissue. Do you see this big gaping hole here? That's dead brain. This is a child who died of something like a massive stroke because they couldn't oxygenate their brain. So a very serious disease. If you are lucky enough to survive it, like I was, you may be left with neuropsychiatric sequelae, which I think I actually do have, but it can be a quite significant event. So this is all bad news, right? Why isn't malaria uniformly fatal? Well, it turns out that after years of exposure, some children, some lucky few, to paraphrase the St. Crispin's Day speech, some lucky few develop protective antibodies. What's an antibody? This is a picture of one. It's a Y-shaped molecule that your immune system makes, and if you look at where my hands are, it attacks invaders with its two arms. And it's extremely specific to what it will bind to. Here's the problem. If you go into a village, the vast majority of kids make antibodies against malaria, but the vast majority of those antibodies are irrelevant. They won't protect that child. It's very few antibodies that are protective. And so that's a major challenge. If we could figure out what it is that the very few who are protected are recognizing uniquely on the parasite, we would have a great leg up to developing a vaccine. Because if we had that knowledge, we could mimic those antibodies in the susceptible kids. And that's really been the focus of our work, honestly, for the past almost 20 years. So here's our approach. We go into an endemic area, and the place we work in Tanzania is one of the most endemic places on planet Earth. The children in that community get more than one infectious mosquito bite per person per day. That's not one mosquito bite per day, which is a drag at your picnic, right? This is one infectious mosquito bite per person per day. It's unbelievable pressure malaria. So we go there, and we follow children longitudinally over time, collecting blood films to see who is resistant and who is not resistant. And then we have to do what I call DNA gymnastics, or molecular biology wizardry. And here's a cartoon of it that'll help me explain it to you. This red thing is a parasite. The golden triangle is a piece of the parasite, a parasite protein, that is being recognized by, remember the antibody? By an antibody in a resistant child. That child is resistant, they make this antibody, it sees the golden triangle. I want to find the golden triangle. The problem is, the vast majority of kids are susceptible. They also make antibodies against the parasite, but they're seeing blue squares. There is a sea of blue squares. You are all blue squares, and I'm trying to find one golden triangle. So that's you. Yes, that's the challenge. That's absolutely the challenge. If we can find the golden triangles, the rest, the biotechnology to turn that into buckets of vaccine is fairly straightforward. If I could immunize susceptible kids with golden triangle, they'll make this antibody and they'll be protected. So that's the goal, is to find the golden triangles. Let me tell you about the study. This was conducted beginning in the 2000s. It was actually funded by Gates to my postdoctoral mentors, Pat Duffy and Michael Fried, who are down at NIH. This is Tanzania. That's the Indian Ocean. That's Lake Victoria. That's Dar es Salaam, Mombasa there. That's the village we work in. Like I said, really is an unbelievable transmission of malaria there. We enrolled almost 1,000 children at birth. And we followed them every 2 weeks with a blood film to measure parasitemia. in the first year of life, and then every month in years 2, 3, and 4. So intensive follow-up looking for malaria in these kids. We have active surveillance. We went to every child, and this is spread out over a 60-kilometer square grid. We went to every child's village every 3 to 5 days to ascertain their health status with a health wagon, with a clinical officer. And despite that intense observation, we still had an under 5 mortality rate of 8%, which sounds horrific, right? In comparison to the children who were not in our study, the under 5 mortality rate is 25%. So it's 1 in 4 children not making it to their 5th birthday. It really emphasizes why malaria is public enemy number 1. We were able to collect almost 50 blood films on these kids, and we used these to decide who was resistant and who was susceptible. This slide is here just to remind me to tell you that there's lots of molecular biology hocus pocus in order to figure out the difference product between resistant and susceptible. And I had an incredible MD PhD student who was on vacation for the summer, and he decided to come back into the lab, and he went through 1.25 million blue squares in order to find 3 golden triangles. So, yeah, I hear it for him. Let's hear it for him. And he never complained. And in fact, I told him the story of Madame Curie, you know, when she had 6 tons of pitch blende delivered to her lab, and after umpteen years she found a microgram of radium. And I told him he got no solace with that story. So he needs to sing in order to get the healing power. Yes, he needs to sing. So we got 3. I'm going to tell you about one of them today. And it was brand new, never been described before, so we got to name it. It's sort of like our child. SEA. PFSEA, Schizont egress antigen. Remember, the schizont is that form just before it ruptures. Just before it ruptures. So we clone this thing. The first thing you want to do, if you're making a vaccine for a parasite, is make sure that the parasite needs that protein. You don't want to attack, say, parasite hair, right? Because what's the parasite going to do? It's going to go bald. And then attacking the hair won't help. You want to attack the parasite's heart. And so we can grow parasites in culture. This is parasitemia and days in culture. If we grow regular wild-type, unaltered parasites, they grow great. If we knock out our gene, which we can do, from the parasite, they don't grow well at all. And so it's mission critical. It's a vital protein for the parasite to grow. That's really good. You want to attack something that's vital. You want to know where it is. So this is micrographs of parasites, the early stages and the late stages. That's just marking their DNA. This is marking our protein. Is our protein in the merozoites? No. In the rings? No. In the trophs? No. It's in the late schizonts, just before they rupture. We want to know a little bit more. I thought this was pretty cool. This is a transmission electron micrograph. This is a red blood cell, and it has been entirely consumed. On the inside, this is all parasite. It's eating the red blood cell from the inside out. That's its digestive, that's like the waste product, that's the parasite poop. These are the developing parasites right there. Now I'm going to take you out to this membrane. That's the outer membrane of the red blood cell, and that's marking it with these big black dots. The little black dots are marking our protein. So our protein is right underneath on the schizont, this is the parasite membrane, and right up on the inner leaflet of the red cell. So it clearly is a parasite protein. And it's a cool picture, I thought. All right, first thing, we immunize mice with our protein. We take the sera from the mice and ask the question, does it kill parasites growing in culture? And that's what this experiment is. These are parasites growing in culture. More parasites go up. 3 different conditions. If we incubate those parasites with nothing, they grow great. If we incubate them with mouse sera before we vaccinated the mouse, they grow great. If we incubate them with mouse sera after we vaccinated the mouse, the parasites grow much worse. 60% reduction in parasite growth, which got us very excited. What if we vaccinate the mice and then challenge them with parasites and watch what happens? So here's parasitemia on the vertical axis, days after challenge. These are 2 control groups. What happens is the mice get high parasitemias and then they die. If the mouse got vaccinated, they control their parasitemias. They go up a bit, but then they stay. The mice don't die. And I'm a huge fan of not dead. This is a survival. Huge fan. Huge fan. This is I'm a pathologist, so I'm supposed to be a doctor of the dead, but I'm a funny one. I'm a doctor of the nearly dead. Like the Billy, the Billy Graham, well, he's not dead yet. Survival. Survival. So they're all surviving. The control groups die on day 10, 8, 10, 12. The vaccinated group goes at least twice as long. This is clinically relevant. If I can keep a child living for another 4 or 5 days, they can get effective drug treatment. So the magic is to extend the survival window. This is the home run, I think. We go back to that cohort of children, 700 children, and we measure antibodies to our protein in all 700 of them, 780. And we ask the question, if you were lucky enough to have made an antibody against our golden triangle, were you protected from severe malaria? Were you protected from all-cause mortality? Were you protected from malaria-specific mortality? This is the number of cases adjusted for 1,500 child weeks of observation. In the black bars are what happened to you if you did not have antibody against our golden triangle. And you can see lots of severe malaria, a fair amount of mortality, and a fair amount of malaria-specific mortality. What's really hard to see, because I had to fake it, is that little bar there. Turns out, if you had antibodies against our golden triangle, there was 0 cases of severe malaria. Absolutely 0. There were 0 cases of malaria-specific and all-cause mortality. This is really exciting. If we could immunize these kids with our golden triangles and make them look like these kids, we would have a phenomenal impact on under 5 mortality in malarious areas. The take-home messages are, one, I should tell you, we've moved this on to non-human primate trials, and it looks very exciting. And so we really are getting ready for phase 1 human trials. Malaria is going to be the great killing machine for the next decade, no question. Multidisciplinary approaches. I didn't even get to talk about the social interventions, drug interventions that are possible, but they're all very helpful and very valid. Bed nets, for instance, just to sleep under a bed net so you don't get bitten as much would be really helpful. But until we have new drugs, because the drugs we have are getting less effective, until we have new drugs and maybe a vaccine, we don't have any vaccines yet for malaria, it's going to kill for the foreseeable future until we get new tools. My hope, my dream really, is that this vaccine, or the ones that are being developed by other groups, I don't want to sound like ours is the only one in town, it's not. But I hope that our vaccine, or those being produced by other groups, will in fact represent at least the beginning, and if not the beginning of the beginning of the end of malaria. Thank you very much.